I used to be the executive director of the Forum for Collaborative HIV Research. However, we have, my disclosures, the outline. I will talk a little bit about the collaborative stakeholder model, regulatory paths for NASH and the hurdles, and then the opportunities for innovation. And just what I was starting to say, we used to be called the Forum for Collaborative HIV Research because that's the only disease area we worked in. But as you can see, we now work in 6 different disease areas. So we're now called the Forum for Collaborative Research. And my talk today will represent the perspectives coming out of the Liver Forum. So the basic forum model is a stakeholder collaborative model that allows process of dialogue and deliberation between government, industry, academia, and community. And the outcomes, deliverables really are greater clarity, understanding, and new research paths that can be engaged in, very frequently also new research funding. And our operating principle is that once a new drug candidate or therapeutic strategy has been identified, the efficient, safe development is in the best interest of all stakeholders, most of all the patients. And we owe it to the patients participating in our trials to really make the best and most efficient use of what the data they provide. So basically we have the different stakeholders, and really where they come together is really what the forum is. It's a very simple concept. Everyone goes and does their own job, but when we come together, it's a safe and interactive space. So we provide information democracy. It's a non-competitive, safe environment. Everyone owns the process. We look for synergy versus duplication, and we really allow everyone to contribute what they do best. So it's a safe space for ongoing co-evolution of science and regulatory guidance. And so normally what happens is that each sponsor talks with each regulatory agency one-on-one, and no one really learns from that experience other than the people at the agency involved in that particular sponsor. So what we do, basically, the forum model is we bring all the sponsors, all the agencies, all the academics together in one setting, and everybody can learn, and it just really accelerates the learning pace and the development pace. So NASH. So we don't really know how to find it. We don't know how to define it. We can't diagnose it, really. We don't have an indication for treatment. We don't even know how to name it, because I'm on the side of people who say non-alcoholic steatohepatitis is really not a very good name to name a disease, because you start off by saying what it's not. But we do know it's an unmanageably huge public health problem for children, adolescents, and adults across the world. So the challenge to industry is to develop new therapeutics in the absence of a realistic and pragmatic regulatory path, although that is changing, validated and accepted biomarkers, and we will talk a little bit about the difference between validated and accepted. And these need to be in the diagnostic, prognostic, predictive, and surrogate endpoints. And we heard something about the context of use yesterday. And we need to also, we don't yet quite have the precise definitions of disease stages and progression. And we don't have a good diagnostic. So if you have a very good surrogate marker or diagnostic that you can use for proof of concept, then you can see if your drug actually works. But if you don't have that, but if you have a drug that really works, then you can really validate a biomarker. But in this case, we don't have either. So we're really kind of groping in the dark, trying to co-evolve diagnostic and therapeutics at the same time. So the challenge to regulators is to develop a regulatory guidance in an area where none existed. And both the FDA and the EMA have been very active and obviously are producing guidances, although they haven't published them officially yet. And it's a very complex pathogenesis. We heard lots about that. We have to look at the short-term versus long-term effects, the benefits and the risks. What is the impact of lifestyle, and what is the context of comorbidities, diabetes, cardiovascular disease, et cetera. And they also need to make sure that they safeguard the health of the public while fostering innovation. So the challenge to the NASH clinical and research community is, when do we decide that it's safe to combine treatment? What is the long-term follow-up versus placebo? What are the ethics and the feasibility of keeping patients in long-term trials when there might drugs be approved already as accelerated approval? And when is it appropriate, for example, to test new drugs in children and adolescents? Back in 2013, the FDA and the AASLD held a combined workshop on challenges and opportunities in drug and biomarker development for NASH. And this was published in Hepatology in 2013. And out of this meeting, this was a 2-day meeting, and you can see that the authors are academic, Arun Sanyal, Scott Friedman, Arthur McCullough, and also Lara Dimick-Santos from the FDA. But what really came out of this was that there was a need for an ongoing dialogue and interaction, that just a one-off meeting doesn't really, it raises all the issues, but it doesn't really solve them. So that then led to the establishment of the Liver Forum. By that time, we had the HIV forum, HCV forum, and CMV forum already in place, so this was the next one. So at one table, we have the regulators, the academics, the societies, and the industry and patients. And it's the same model that I've already described to increase clarity, efficiency, innovation, and decrease uncertainty, redundancy, time, and risk. And so again, it's a cross-Atlantic regulatory and industry academic perspective. And we also, just because of the informal infrastructure, there's a lot of infrastructure for coordination across efforts among the Liver Forum members and partners. So we've heard a lot about the process of disease and the slow nature of getting from the normal liver all the way to the hard outcomes like death and transplant, HCC. And so how do you develop a drug in this context if you can't really predict what's going to happen? So we have baseline, we then have a natural history that happens a little bit differently for each patient, and then we have a clinical endpoint. And here we need to find a diagnostic and prognostic biomarkers that will allow us to include those patients that are likely to proceed to here. But this, of course, can be many, many, many years. So what are surrogate markers then? Surrogate markers are biomarkers, and they can be serum biomarkers, imaging, physiological, et cetera, that predict this. So surrogate markers, we could have one that's sort of at mid-stage of disease that predicts that, or maybe it would be more efficient to just enroll patients at the very late stage of disease, or even patients early on in the disease that you could predict that they would get to maybe advanced fibrosis, and then we already know that advanced fibrosis would lead to clinical endpoints. So, and Arun, I think, did a very good job yesterday of sort of describing this quadrant of the intersection between fibrosis and the disease activity, so disease stage versus disease activity. And of course, that will all play into that in terms of what we pick as a surrogate marker. I think this audience is very familiar with the two scenarios of accelerated approval, which is based on a surrogate marker that's reasonably likely to predict, but that needs to be followed by traditional approval. And once this particular surrogate marker that was reasonably likely to predict has been accepted to predict, then that can become the primary endpoint. So NASH right now, we're here. So whatever we use as a surrogate will still have to be validated against the hard clinical endpoint. And so the hard endpoint is the clinical benefit, and I think this audience knows all of that very well as well. And for example, some surrogate reasonably likely to predict clinical benefit would be complete resolution of steatohepatitis and no worsening of liver fibrosis, at least a 1-point improvement in fibrosis, and so on. But this will also depend for each drug and disease combination. And as we already heard, NASH is really a complex of pathologies that interact. So a validated a surrogate, once that is accepted to predict, then that can be used for regular approval, and that's of course where we want to get to in the NASH setting, but we are not there yet. So what we're really doing is we're learning while we're doing, and we're doing while we're learning. So the regulators and industry and everyone is learning at the same time. And I'm just posting some slides. These are on our website. This is from our EMA representative. Since I've talked a lot about the FDA, I thought I would show some EMA slides, where Elmar Schabel basically told us about the latest updates from the European perspective. And he had already shown this particular slide previously. So, but again, kind of bringing into play and into context the changing evolution of knowledge that we have and how that will affect how we interpret endpoints. And for example, the previously proposed presented co-primary evaluation of the composite endpoint has been accepted by EMA. And he goes through the rationale for why they have accepted that. But now the intended scenario, what he adds in 2017 is that the intended scenario with conditional licensing accelerated approval will be more difficult in later development programs with regard to the fulfillment of the condition of the phase 4. So how can the interim evaluation account for different mechanisms of action? And that's, again, we're learning while we're doing, and we're doing while we're learning. For example, coming out of the Centaur trial, that depending on the mechanism of action, the exact definition of what your surrogate is may need to change. And so the composite of NASH resolution and no worsening of fibrosis may not be sufficient. And so we may be we may need to be able to show an independent effect on fibrosis. So again, the addition in 2017 that the effect should be shown on the individual level. So here again, talking a little bit about some of the recent findings. The resolution of NASH is not an appropriate endpoint, see Centaur trial, which I had just referenced. And the change in character of inflammation may or may not be demonstrated. How can a sufficiently strong interim endpoint be established? So what I'm trying to show you here is just the amount of consideration and discussion that goes into these decisions. And if the agency had to do this individually with each sponsor, we would never get to the answer. But we can now do this together, and everyone can learn from each other while we're doing that. So these are posted on our website. I'm not going to go through all the details. I just wanted to give you a flavor. So the challenge basically then is, at the moment, the validation of a surrogate endpoint requires validation. And to go back to this slide, we want to go from here to here, but really just be able to use these surrogate markers to predict and have those be validated. So we heard yesterday again about the context of use, and I like to divide the biomarkers and endpoints into disease-focused and response to intervention. So for disease-focused, we need to diagnose and we need to have prognostic biomarkers. How quickly is that patient going to advance in the absence of an intervention? So it's basically natural history. And then for the response to an intervention, we have the predictive, the pharmacodynamic, and the efficacy biomarkers. But in NASH, we're at the point where we really don't really understand what happens at baseline. Because it's a very messy disease. So the Liver Forum, we really decided, let's first focus on this, because unless we know what we're talking about at baseline and measure that precisely, how can we measure what the outcome is if we haven't measured the beginning precisely? So that was one of the first things we focused on. And we have in press, or just published, one of the outcomes, which is case definitions for inclusion and analysis of endpoints in clinical trials for NASH. And so in this article, that's an outcome of many, many, many discussions amongst Liver Forum members, and you can see the working group members here that were included. And of course, many other members also participated in these discussions. But it's really now we have a framework for, basically we took whatever the science is now, is how we can define it. So, but now we can go to integrating new diagnostics and biomarkers as the, sorry, I keep clicking the wrong one, as the science evolves. And we also then can use the same framework to then define outcomes, such as NASH resolution. We can facilitate cross-study analysis, and we had a working group that put together baseline parameters in clinical trials that was just published as a commentary in gastroenterology. And we also, as Arun mentioned yesterday, we work on coordinated biomarker development where we have FDA, EMA, the NIMBLE network, and the LITMUS network in Europe, NIMBLE here, and drug developers and diagnostic developers, and provide the overarching support for this coordinated event. So innovation. So one of the big things at the FDA now is the totality of evidence, and that means basically integrating evidence from multiple sources such as RCTs and real-world evidence. And Target NASH that we heard about yesterday and NASH CRN are obvious examples of this. So one thing we're doing at the Liver Forum is also establishing a placebo arm cohort which can complement those other efforts, and hopefully that can reduce the need for long-term placebo exposure, which should facilitate recruitment into trials. So tackling challenges through innovation, again, adaptive trial designs is something that we've been discussing. And there's two ways of doing that. So one of them is seamless trial designs where you move from one phase to another without hard stops in between, or patient enrichment trial designs, dose adjustment and randomization probability. So these are all different types of innovations we can apply. And the potential impact, if you look at the traditional development path where you have distinct phases that have very hard ends and stops and beginnings, we can really think about it more as a continuous process that can adapt to a more diverse, heterogeneous patient populations, and then we pick those that really respond, and then we can actually, in the end, pick a population that we can really show the effect in. So, and hopefully we can reduce the cost substantially. So we're starting from the ground up. We need a common language, and we've been working on that. We need common elements. And basically, if we take all of these building blocks and look at them carefully and work collaboratively, we can actually start a structure. So this is obviously really very much informed by the Liver Forum deliberations, and any opinions I expressed are my own, and do want to acknowledge all of the financial sponsors of the Liver Forum.